everybody. We got the first here of two biology presentations tonight. First is Chloe Oakland, outgoing senior from Albuquerque, Minnesota. This is actually the second algae project that we've done. Didn't do a presentation for the first one because, long story short, we just learned that FLIGL can't keep water fleas alive in the lab. So we're going to do the second one where we actually work with wildly caught species. And that, just let me take it away, right?
Yeah, so my project's on water quality and algae of the Iowa creeks. So we found that the most common algae that is found in Iowa is the blue-green algae and cyanobacteria. I've got some of the blue-green algae that is pictured there on the left and cyanobacteria here on the right. So algae is basically simple plants that is found in many different types of waters, such as like lakes, streams, rivers, ponds, and moist soils. And then it can grow in many different types of forms, from being single-celled to multi-celled. And in the multi-celled, they usually grow in like strands or colonies, but they all contain chlorophyll. So cyanobacteria were in the past referred to as blue-green algae because of the way that it appeared in the water. The dense growths turn the water green, blue-green, or brownish-green. And cyanobacteria are relatives of bacteria and are photosynthetic, so basically they can make their own food. Blue-green algae, it's actually opposite, referred to as algae, but not actually algae at all. And it's actually a type of bacteria called cyanobacteria and thrives in the warm, nutrient-rich waters. So it's more prominent in the late summertime, and it can make water bodies smell pretty badly. And despite its name, it actually contains blue or red pigments, I found. So the good and bad. So algae is actually good if it is kept at the base level. It provides oxygen, captures carbon dioxide, and it can keep the aquatic life pretty healthy. It's the base of many of the food webs, so a lot of different types of animals in the water can feed on this. But if it actually grows out of control, then it can turn pretty bad. This is a common thing that actually happens here in Iowa because of all the nutrients that come off of our fields and that gets in the water. Eutrophication is very common here because of those nutrients getting into the water. The algae, when it grows out of control, excess nutrients come off of the fields. The algae it can cover the whole surface of the water and produce toxins that harm the aquatic life. And when the algae dies, it uses up a lot of the oxygen, which leaves less for the life in that water, leading to fish kills and many other problems. So basically, it's good if it's kept at a base level, but pretty bad if it grows out of control. The three most common factors that affect the algae growth: nutrients, temperature, light. There's multiple other types of factors too, but these are just the more common ones that I found. So nutrients, they get in the water again in many different types of ways, but in Iowa the main cause is the fertilizer runoff. So the water makes its way from the farm fields into the water, and then also secretions from wildlife is common. Too much of the nutrients, not good for the water again. Like I talked about in the previous slide, eutrophication happens. This is why the stormwater management system is pretty important. I got a picture here and it shows a couple of different types of ways that this happens. Temperature is also very important. Certain algae grows better in the cold, such as diatoms and green algae, while other types, such as cyanobacteria, prefer the hotter temperatures. Usually the population of the water is more diverse in these colder temperatures as well. So light, very important as well. More light leads to algae growing faster, but too much of the light can hurt the algae. And since cyanobacteria prefer the hotter temperatures, they actually have a built-in sunscreen to protect them to the sun, but that's just to an extent before it actually becomes too dangerous. Nitrate can act as a nutrient for the plants in the water, but too much of this causes problems, which is a common thing here in Iowa as well. It's released in the form of a salt, so sodium nitrate, potassium nitrate, and ammonium nitrate, and these will quickly dissolve and liberate the free nitrate ion. Since nitrate does not bind to the soil, it's easily washed away into the water. Some of the runoff from fertilized lawns some of the most common sources of nitrates in the water include things like the wastewater treatment plants, failing on the septic system, runoff from animal manure storage areas. Too much of the nitrate can accelerate the eutrophication, which causes an increase in the plant growth and even change the types of plants and animals that live in the water. So here I have a graph. So you can see, like, the 1950s to the 1980s, they started using a fertilizer that was very rich in the nitrate levels, which is why there is such a drastic increase in there. About the 1980s, they figured out what this was doing to the water, so they decided to cut back on the amount of nitrate. There was still nitrate in that, in the fertilizer, which is why it's still growing, but they have cut back a lot. So the materials that I needed 8 collection jars for the different creeks that I went to, label tape and marker just to stay organized, section microscope and a regular one, Segwick Raptor counting cell, I don't have pictures anymore, pH, nitrate, phosphate, turbidity testers, gloves, and some sanitizer. So a sample was collected from each of the streams, it was labeled and brought back to the lab to study more. Gloves were worn at each of the streams and sanitized to prevent any cross-contamination from happening. Once back at the lab, each sample was looked at under the dissection microscope to see if there was any macroalgae in there. They were then tested to find the pH, nitrate, phosphate, turbidity levels. We used this picture here on the left. Those were what we used to test those levels. And then using a Segwick Raptor counting cell slide, a sample was looked at and sample was loaded onto it and looked at under the microscope. We did a transect line across the horizontal axis of the slide, and specimens were pretty much counted and distinguished to record and put into a Shannon index calculator. I used the Statology website for this, and then using the PRISM software, graphs were created to find species richness with the barn distance and the crop field distance. A log Shannon index was also used on the graph with the barn and crop field distances. And then using the software as well, the T-charts were created to find the log Shannon diversity index with the high and low nitrate levels. These are some of the different types of stuff we found in each of the different creeks. So we want to notice how each of these pictures are very different in the number of each of the species being shown. These each have 4 different species of trees, so this shows species evenness within each of the communities. Species richness is also shown in the amount of each of them that there are. Each of these can be incorporated into a diversity index, and we use the Shannon Diversity Index, which is the most common one to use. I added a little map of all the different types of creeks that we use, but we used these 8 that are shown here. Then these are the results for the crop field distances. The left here we got the log Shannon diversity one, and we got the species richness on the right. Our analysis was not able to detect enough of the significance between the X and the Y, but there appears to be a pattern, so on some of them, so better analysis could be done in the future with some more sampling. And then this shows for the barn, our analysis again was not enough to detect a significance between them. And then these are the charts. They also did not show enough difference. Normality tests were ran. We used a Farrow will test for this, and species diversity did not come back normal while running a normality test, so we did need to transform it using Excel log 10 x1. We found that no macroalgae were found in any of the different samples. The pH was 8 in each of them. Phosphate was 2 in all of them besides Elk Creek, which was 1. Turbidity was 20 in all of them, and again, Elk Creek was the only difference with 40. Elk Creek needed a second trial as well because we did find some Daphnia in that first sample, and we wanted to test to make sure that it wasn't affecting anything in that one. Combs Creek was not used in the plotting for the crop fields or barn distances just because it did not have, it may have been an outlier, so no species were found to get any information found out of that one. And a diversity index was needed to be transformed. For future research, we could use more sample sites, know the fertilizer used in the nearby fields, just to know how that's affecting the nitrate levels that are in the water. We can look into how algae differs during different times of the year due to weather, since that does affect the algae, the diversity of the algae. We can do more quantitative measurements for nutrients, more transects, know the percent of agricultural land near the creeks. And then these are all my works cited.
Anyone have some questions?
I'm ignorant of most of your vocabulary, but are those results good, bad, or indifferent?
They definitely were not what we expected them to be. We thought that, like, we thought there would be more of a line for them, so. Sure.
Yeah, can I follow up on that? Could you go back to the slides with the graphs?
Yeah. This one or this one?
Yeah, any of those. Yeah, you're right, that there's no significant differences here, but you said there were some patterns. What patterns do you see you thought were important? That might help with your question.
This is like, I'm not sure.
So maybe start by explaining what is crop field distance.
Oh, the crop field distance is basically just the distance from our point of sampling to the point of the nearest crop field bed. And we just measured that distance.
Okay. And so how would that correlate if it had turned out to be significant with species richness?
I'm so sorry, what is that?
How does species richness change as crop field distance changes?
Oh, we just figured it would go like more of a, line up. We found that they were more all over the place instead of more of a straighter line.
Chloe, could you go to the next result graph, because I feel like that one is
This
a
one?
little bit, yeah, that one
Yeah.
seems like there's some clustering
That's
things there.
stronger.
Yeah.
With the barn distance, which, is that what it sounds like it is?
Yeah, that's the,
Okay.
that's the one that has more of a, progression.
What do you think the barn has to do with increasing diversity index or increasing
We
species?
wanted to look at
distance?
like the barn distance because of like the animal manure, that would bring like the nitrate levels up, and that's kind of what we were measuring.
Leaking out of the barn into the street.
Yes, yes.
Are these
Like
in
in
two different places?
Huh?
You got two graphs here. Are they, how do you
Yeah,
differ
the
from
first
each
one
other?
there is the log Shannon diversity index, and this one is the species richness. and they're both, on this one for both the barn and crop fields, ones for these.
Okay.
Do you think having a larger sample size would have helped your data?
Yes. I believe it would have. We only got 8 of them, but it probably would have helped to have a lot more than that, yeah.
I'm like you, I don't know all this stuff and the terminology and stuff, but when, sometimes in the summer, our Waldorf Pond is just gobs of gross algae going
Yeah.
into there. Is that from the fertilizer on the grass, do you think?
It could be, yeah. I mean, the fertilizer does impact the algae that's in the water, yeah.
So what should we do?
There's different types of fertilizers that don't have as much of the yeah, certain fertilizers wouldn't impact the water as much, but you can also do certain things like around the waters, like on, what is up here? Like, some places use these different things to keep the nutrients out of the water. But I'm not sure on the pond out there, but there's different types of ways to keep the fertilizer out of it.
There's a lot of inputs going in that pond with no outputs. So besides the fertilizer, we got dead leaves and collected air vegetation. And it's just a very small system. Could
Are
easily
you
overwhelm.
familiar with the concept of limiting nutrient?
Not fully, no.
So the one that is present in the lowest supply determines how much growth can happen. So is nitrate the limiting nutrient? Is everything else more abundant?
Guess I'm not exactly sure on that. I don't think nitrate's like necessarily like the only thing that goes into it at all though.
Okay.
Phosphates are more or less equivalent across the sites.
Phosphates?
Yeah, they're more or less equivalent across the sites.
Oh, yeah. So nitrates vary a lot more than phosphate?
It is.
How did that?
Yeah, we found that if I can find it again. Just in ours alone, we did test phosphate and there wasn't as much of a difference between just for our sites.
So you could have had a huge variation in nitrate, and if it's overabundant and the real controller of growth is phosphate, it would have been hard to see change in
Yes, so now we have the aquatic sequel. Tyler is on the baseball team here at the university, he's a senior as well. We also had some interesting challenges, to say the least. We suspect some of the raccoons got a little too curious with some of the traps at some points. Also, I had to start questioning sometimes if I got some of Paul's frog traps by mistake or something, because one of the sites, it was nothing but like dozens of frogs in the trap the first time we collected it. It's like, I'm questioning myself at that point. But yeah, so he looked at,
No, at least they weren't bullfrogs.
they were not.
I hope you shot all of them down.
So, kind of a little flip though, we're kind of going from the micro to the macro here, looking at fish, and I'll let you take it away, Tyler.
Yep. So, sorry for anyone that didn't understand all of Chloe's, because mine's gonna be pretty similar. Here we're looking at factors affecting fish diversity and size here in North Iowa. So with aquatic diversity, exploring diversity is beneficial in understanding how the health of ecosystem is. Typically, having more biodiversity in an area indicates that it's healthier and more productive. With aquatic diversities, limited to nutrients available in the water, oxygen, phosphorus, nitrogen, silica, and many others. Here in North Iowa, we have the Winnebago River watershed, which was what we were looking at, covering the 4 surrounding counties. Within that, there were 10 lakes that we also looked at. Some of them are included there, but we'll talk about those in a minute. Kind of similar here, we're looking at agricultural influence. So obviously has a big role here in the Midwest. Fertilizer, pesticides, some of the main contributors specific to changes in aquatic habitats. And so by being able to determine the overall effect this has can be the first step in improving aquatic systems, ecosystems across the state. So specific to North Iowa, our goal here was to catch and measure small minnows and darters within the creeks of the watershed here. And then using that data, in addition to nearby farmland, the idea was to determine what factors, if any, had an impact on species size, species composition. So for our sites, I had the same 8 sites as Chloe, but this was from Colmus Creek, and they were all relatively similar to that. Elk Creek was quite a bit bigger, but for the most part the same size and water flow. Before any collections, we had to buy a fishing license, and then the IACOOK was filled out and approved. That was just for to ensure like the animal safety. But with our study, we were just catching and releasing them, so it wasn't really an issue for us. And then of the 8 sites, 2 of them were removed because of minimum water flow. And then at each of the 6 remaining sites, we had 2 of these traps at each site. And so that was kind of just the one half of the trap, and the other half goes on top. And there's like funnels on either side that the fish swim in and can't swim out of. Inside there is our dog food bait, which is what we used. With the traps, before each trapping day, they were all sprayed with a 10% bleach water mixture and then rinsed with water. We did this to eliminate any possible contaminations across one site to another. And then for each trap, we had the dog food there, which we just put in little mesh sacks and zip tied them up and put them in there. So. And then we used nylon rope to secure the traps, and they were all bought new, so there wasn't any concern of contamination there. So our first site, Buffalo Creek, pretty unproductive, but We did catch one black bullhead. The rest of the other two trappings weren't ideal, as some of the other sites were the same. So we did catch one here. Pike Run, these were the frogs that he was talking about. So I actually got really excited when I went to pull them up and saw a bunch of stuff moving around in there, and turns out it was just 57 of these leopard frogs. Wasn't ideal, so we didn't put the traps back out there again, just to minimize catching any more of them. Elk Creek, another frog. That was the issue that we faced a lot. But this one was a bullfrog. Only one of those was caught. That was it for that site. Comus Creek, a little more productive. These were the creek chubs. One was 15.5 centimeters. The other little guy was 3.5 centimeters. Here was the most productive, still not ideal, but 6 more of the creek chub on the right there. And then we caught 2 of those are Johnny darters. We caught one, that one was 6.4 centimeters, and then there was just another little guy that we caught there too. Bear Creek, this one just outside of town by the golf course. First trap we caught 2 black bullheads. These ones were pretty big, 15 and 11.5 centimeters. Then the other 2 times caught 1 shiner each. So that one was at least somewhat productive all 3 times that we put the traps out. So switching from this lake diversity, because of our limited number of fish caught, we decided to kind of add to our data by using the DNR website, the natural resources. They had a data set that provided an in-depth overview of all fish species across all lakes here in Iowa. And then with this data, we wanted to look at nearby farmland of these lakes to try and determine if or what factors agriculture has on the aquatic species. So for our data collection with this, Google Earth was my best friend. I don't know if I like it anymore, but so for each cardinal direction, we measured 5 kilometers each way, and then found out what percentage was farmland. And then, so for each lake with the DNR numbers, that was from 2022 to 2024. And then with that, we used Statology to calculate the same Shannon index, and then GraphPad Prism was used to calculate our data. And then with that, we used the same normality test, and our data was shown to be within the range of normal, which I don't know how that happened, but it did. So those are our numbers there. So our results, I mean, here they look as we would expect them to look with, well, maybe not as we expect them to look, but this would show that having less ag land surrounding the lakes would increase the species richness and the Shannon index. But with that, the point on the left for each graph, that was Clear Lake, and it had a significant amount more of non-farmland. So we ran the data set again without that, and we got this, which did show, in the computer it said it showed a trend, but I mean, it's hard to tell with increasing ag land and increasing species richness and the Shannon index. So that would indicate that more ag land would have more species diversity. Yeah, with Clear Lake is also drastically larger than all of the other lakes. So I mean, that obviously had an impact. As population, or as habitat increases, population's gonna increase, which the diversity is also gonna increase. And then with that, some of the lakes are also more popular fishing sites, so more stocking efforts and larger pressure from fishermen is gonna have an impact on those numbers. So we tried to go away from, they had stock numbers and then like an intermediate, that was their classification for the fish size. So we went away with the stock size and the trophy size just because of the pressure that was put on them. So with that, future research would definitely be beneficial in this. We didn't find any clear trends, but still got some good useful information. With more time and resources, could have been possible to place more traps along more locations. Ideally, going into this, we were hoping to catch around 100 across 8 sites, but didn't obviously work like we wanted it to. And then increasing the number of trapping days earlier in the year. So these were all done end of October, beginning of November. So I mean, the colder weather also has an impact on the fish in the streams. So they tend to go more into the lakes and deeper waters to get away from that. So being able to go out when it was warmer could have helped us, I guess. So, but yeah, that's all I have. So if anyone has any questions.
What kind of dog food did you use?
Just Dollar Tree. Nothing special.
Yeah, the Clear Lake had quite a profound effect on the
Uh-huh.
results, didn't it?
Yeah.
Were all your transects always 5 kilometers?
Yeah. So we,
So did you account for different sizes of your lakes? I mean, did you control the proportional differences? Because the larger the lake, the less ag land you have to compare to.
So with that, I just started my measurement from like the farthest north point of the lake and measured 5 north from there, and then the farthest south and went from there. So it was not ideal, but with the situation, that was what we thought was going to be
So
the
it's
best.
5 kilometers from the edge.
Yes.
Gotcha.
Yep.
Your list of sites? Looking at other creeks, did you include stream flow velocity as a factor to pay attention to?
No, we didn't. Initially, we were going to look into like sunlight, temperature of the water, but we kind of went away from that and kind of just focused more on. With this, we were hoping to find more fish, so we were going to compare that also to nearby farmland. But just because of the small number of catches, we kind of went away from that. But we didn't measure anything like that. Yeah.
This time of year, what could cause a mass congregation of frogs?
That's what I've been asking myself.
There was a lot of discussion about that while we were driving around.
I mean, honestly, I'm not sure. I don't know if it has something to do with them cutting all the corn, or if it's. I honestly have no
Do they
idea.
like that dog food?
They must like dog food, yeah.
The big cover should be jumpers. Those fish are really tiny. I was surprised, like, with the gloved hand, and it's like this little tiny thing. Is that because the holes going into your trap are so small, only the tiniest can go in?
So on the far left there, I don't know if you can see it, but they're only about 1.5 inches, 2 inches maybe in diameter for them to enter.
So what does that do with your data if there are larger fish that can't get in the trap?
That is a good point, yeah. So obviously, we're not going to catch fish that are very big. So I mean, we were focusing more on like the smaller fish. So I mean, it doesn't have as much of an impact, but it definitely could have, yeah.
And would those be full size, or those are babies that are just being born?
So some of them are full size, like here, that one's around full size on the top. with the catfish that we caught, the one on top there, that one's, they can grow to be very big, so. But yeah, for the most part, and then that one on the left, that one doesn't get more than 10 or so centimeters typically.
If your farmland factors have an impact, would you expect it to be stronger on the adults or on the minnows?
I would say the minnows, just because they're still in that growing process, and so I mean, if there's more nutrients getting into the water, there's going to be more algae and stuff, which would theoretically increase the size and the diversity there. So that was, I mean, that was kind of our whole point, was to see if that had any influence over the overall ecosystem there. Yep.
Do you have any information on all the vegetation at the separate collection sites?
Amount of vegetation?
Yeah.
No, we didn't have anything like that. With this one, I mean, they were all pretty similar. Just mainly grassy areas, right around the site. For all of them except for Elk Creek, they were pretty much running through farmland, so.
So one of the factors contributed to all the frogs on the one site.
That is true.
More frog habitat.
Yeah.
Did you dispatch of that bullfrog and found it?
No, unfortunately.
I'm curious, could you catch a crayfish?
Yeah, that's, so before this, I've used these traps before, but it was only for crayfish. I've never used them for catching fish, but, so.
How many crayfish did you catch? If you can.
That wasn't part of this study, but.
No, but I mean, in this study, you did catch a crayfish.
No, not in this study.
Oh,
But I was just saying before this, that was, I had used
okay.
those previously.
Gotcha.
Yep,
Any signs of invasive species?
yeah. The frogs.
Well, the frog. Okay. Sounds good.
They're not invasive.
Maybe these ones are.
Northern
There was
leopard
I think it was the
That's the
bullfrogs.
invasive
Yeah,
kind.
yeah,
That's why I was hoping to dispatch them.
yeah.
And bullheads aren't invasive?
The catfish, no.
Yeah, sure.
They're native here, as far as I know.
Okay. That's just good.
Any more questions? Thank you so much. Thanks to all of you who came to listen to wonderful presentations